All right, today we're diving into one of the most incredible, most important processes in all of biology. We're about to unlock the secrets of Prophase 1, the tiny little event inside our cells that is literally the reason you are, well, you. So, you know how you look in the mirror and you can see your mom's eyes, maybe your dad's nose, but you're definitely not just a 50-50 split. You're not a carbon copy of either of them. So where does that unique blend, that thing that makes you, actually come from? Well, the answer is hidden deep inside our cells. There's this amazing microscopic process that's kind of like a MasterCard dealer, perfectly shuffling the genetic deck just before a new life begins. And that's exactly what we're going to get into right now. Okay, first up, let's talk about the Great Genetic Shuffle. Now, when you hear the word shuffle, you might think of something random, a bit chaotic. But let me be clear, this is anything but. It's a beautifully choreographed, incredibly important event that creates genetic variety. So where does all this action take place? It all goes down during a phase called Prophase 1. This is the main event in the whole process of meiosis. You know, that special kind of cell division that makes sperm and egg cells. And get this, Prophase 1 is the longest and honestly the most critical part of the whole show, because this is where all that genetic variation is actually created. But before the shuffling can really kick off, we've got to meet the key players, right? So let's set the stage and get to know the stars of this whole process, the chromosomes. First up, we have what are called homologous chromosomes. The easiest way to think about them is like a pair of cookbooks. You get one from your mom and one from your dad. Now, both cookbooks have all the same chapters, a chapter for eye color, a chapter for hair type, and so on. But the actual recipes in those chapters might be just a little bit different. Now, here's something that's super important not to get mixed up. Homologous chromosomes are not the same as sister chromatids. If the homologous chromosomes are the original cookbooks from your parents, sister chromatids are the perfect, identical photocopies you make of each one right before you start cooking. That difference, original versus exact copy, is absolutely crucial for understanding what happens next. And now for the main event. It's a process so elegant and coordinated that we're calling it the crossover dance. This is where those homologous chromosomes, one from mom, one from dad, partner up and get really, really close. So here's how this dance goes. First, in a move called synapsis, the partners find each other and line up perfectly side by side. Then comes the big move, crossing over. They literally reach over and exchange pieces of DNA. It's just like ripping a few recipe pages out of one cookbook and swapping them with pages from the other. Finally, you can actually see where this trait happened. They form these X-shaped connections called chiasmata that hold the pair together. And these chiasmata are so critical. They're not just some random leftover connection. They are the physical proof that a genetic trait has just gone down. And they physically hold the chromosomes together, making sure everything is aligned perfectly for the next steps. You can think of them like the clasped hands of our two dancers, locked together after making an exchange. So after all that intricate dancing and swapping, what's the big result? Well, it's a brand new, never-before-seen combination of genetic material. I mean, think about it. Before this dance starts, things are simple. You've got one chromosome that is 100% from your mom's side and another that is 100% from your dad's side. They're totally separate. But after crossing over, wow, the result is just incredible. The chromatids are now genetic mashups. Each one has a completely new mix of genes from both parents, all on a single strand. They're not just moms or dads anymore, they are something totally new. Okay, so we've seen the how, but let's zoom out and talk about the why. Why does this tiny little shuffle matter so much? Because the implications are absolutely massive, not just for us, but for pretty much all life on Earth. So if you only remember one thing from all this, let it be this. Prophase 1 is basically the engine room for genetic variation. By creating gene combinations that didn't even exist in your parents, it becomes the foundation of diversity for an entire species. Oh, and remember those chiasmata we talked about? Turns out they also have a really practical job. They make sure these new, shuffled chromosomes get sorted out correctly in the next stage. To put it as simply as possible, this one phase, this complex little biological dance, is what makes every single one of us, from an ant to a whale to you, a genetically unique individual. It is the fundamental reason for all the incredible diversity of life we see around us. And that leaves us with one last kind of mind-blowing question to think about. I mean, if every baby was just a clone of its parents, how could a species ever adapt to new challenges? like a new disease or a change in the climate. It couldn't. So you have to wonder, without this incredible genetic shuffle, could complex life as we know it even exist at all? It's pretty amazing to think about.